We're going to change gears a bit again now and go to something that we see as one of the most fun things we do at this meeting is our OIS Innovation Award.
When I was on a committee with Jude Samolsky, another co-founder of the company, Jude's thesis was on AAV as a vector. It became clear to me, working in AI already, that it's an isolated organ, it's small, and it was clear that very soon there were going to be genetic causes of blindness understood, and it was obvious it was the way to go. And so we started doing this, and the company began in 1999.
Pretty much all of the initial work in orphan ophthalmology and AAV gene therapy, somebody was collaborating with Bill. Bill would design the vectors, they would be doing some of the experiments, and so we had quite a bit of animal data that then supported us in getting the true venture round of financing. And that early animal data was supported through some of this early seed funding we got, through the SBIR program, and also from the Foundation Fighting Blindness, who funded not only Bill's lab, but also AGTC in the early days.
If you're looking at recessive diseases of the eye, dogs are a great model because everybody wants their dogs to look alike. And so all dog breeds are inbred, and many of them have retinal diseases that are very parallel with human diseases.
And we use a lot of those animal models in developing our preclinical package.
They're much more useful than mice because they have a human-sized eye. And if you know about dosing, you know that dosing from a mouse to a human is virtually impossible. It's a guess.
How did you first were able to study it in humans? How were you able to get that done?
So it was really developing the safety package. And you know, that did take, during this initial time period, quite a long time. However, you know, I do have to give due credit to the FDA and the regulators that we were working with, in that they very much understood by this period of time that vectors are different. You know, an adenovirus vector, an AAV vector, a lentiviral different. They are all different from each other. And so we had to put together a fairly comprehensive data package, but when we put that together, we were sure to test significantly higher doses than we were going to use in the clinic. In our very first product that went into the clinic, the toxicology doses were actually 480 times higher than what we planned in the human clinic. So we were really pushing the envelope of providing everybody comfort on the safety level.
Can you talk a little bit about the clinical trials that you're in right now to give the audience a sense of the different programs that you're doing?
So right now we have the in-life clinical trial is the LCA trial, and we're waiting for the 2-year follow-up data from that trial. And then next year is going to be an action-packed year for AGTC, and that will have 3 different indications in the clinic. One is for X-linked retinoschisis, which is a disease that affects a protein that holds the layers of the retina together. Another is for a form of achromatopsia, which is a disease where there is no cone photoreceptor function. The cones are there, and we now know that for sure because of the adaptive optics technology we're using, but they're just not functioning. And so that trial will be in the clinic. And then also that Alpha-1 project, the one non-ophthalmology program we're working on, we're going back with a new delivery system. So we'll have 3 clinical trials ongoing. Because of the safety that we've been able to build up over the years, the comfort that people have with the safety of AAV vectors, we are going to be able to begin these trials at a dose that could be effective. We're no longer in the situation where we're having to start at very low doses and gradually work our way up. So we're very hopeful that we'll be able to have some very positive data coming out of these clinical trials.